So Heather, do you want to mention something?
Sure. Well, I just look at the proteins that are required for cells to get into the central nervous system and allow them to become pathogenic in our mouse model.
I have an interesting theory that some of these T cells that induce disease in the central nervous system must traffic through the gut or the intestinal system before they get there. We have some reason, we know where the T cells develop, we know they develop in the lymph nodes, but it takes some time for them to travel from the lymph nodes to the central nervous system and induce disease. And we really don't know what happens during that period of time. We know some of the factors that are required for them to induce disease, so when they leave the lymph nodes, they're not pathogenic. Something has to happen to them to make them pathogenic. And I have some data from some animal models where when we track cells that we have specific markers on, we can see where they go. And I have data to show that in between the time they leave from where they develop to when they end up in the central nervous system, that they actually end up in lymphoid tissue that surrounds the intestine. So we call them Peyer's patches or the mesenteric lymph nodes. What happens to them there and how they become pathogenic there, we don't know. But some of the markers that cells typically upregulate that allow them to go to the intestine to fight pathogens within the intestine are also expressed in cells at the onset of disease when the cells get into the central nervous system. So there may be some link there, and that's currently what I'm working on.
And it's interesting because, you know, there's a lot of questions about how diet can affect MS. There's some data going on now about worms, parasitic worms in the intestine, and how that may influence MS. So I think Stephen's research speaks to that because as these cells travel through the intestines, they may be modified and we may be able to stop them from becoming dangerous through dietary interventions one day or using parasites even. And Josh?
I just started, but I'm looking at the way that cells from the bone marrow really gain access to the brain in terms of interacting with a very prominent cell type in your brain, more prominent than neurons, in fact, called astrocytes that make up the blood-brain barrier that gives your brain that sort of special kind of protection from the immune system that that the rest of your body doesn't have. So I'm looking at how factors that immune cells make, proteins that immune cells make, kind of allow them to get through that barrier in one way or another, and how losing some of those factors changes the way the disease progresses. In the animal model, we use the EAE model.
I just, like you said, I just came from California last month, and where I finished my PhD, and I was working on multiple sclerosis as well, and I was working on stem cells in the central nervous system, in the brain and in the spinal cord, how to activate them and how they migrate in the central nervous system. And coming here, I'm just starting some projects where we're going to look at kind of bridging immunology and stem cell biology, which, as many of you guys might know, in the field, there's like the stem cell side, there's the immunology side. The stem cell is trying to help regeneration. The immunology side is trying to help prevention. And hopefully we can bridge the two. And the idea is to do both at the same time. So we're looking into projects that'll look into that.
Okay, thank you, guys. I'm taking them out to lunch afterwards. So. Just to show you, so some of the work that Josh and Heather are doing has inspired us to think of new types of drugs to block the immune cells from getting into the central nervous system. You may know that that's how Tysabri works, which is an IV drug that's given once a month and has been very effective in decreasing relapses. We're trying to develop a new generation of Tysabri that may be even more effective and have less side effects. And we've actually made one such molecule in mice and used it in mice, and it looks like it's working quite well. So we'll have to see if it works in humans. Actually, here's a diagram showing how that drug works in mice. This is the degree of disability over time in the animals that get the MS-like disease. And here's a control group that was not treated, and the animals that were treated, you could see that they were protected. They're not getting disabled. So we hope one day that's going to translate into a new drug for people. And here's some data that we have. This is actually showing the optic nerve, the nerve that transmits signals from the eye to the brain that's affected in multiple sclerosis during optic neuritis. And with Dr. Geiger, and this is a project that Kevin's starting on, we can damage the optic nerve so that the nerve fibers die. But then we are looking into methods to stimulate regrowth of the nerves. And this is actually using a certain product derived from fungus. And we and others have found that if you introduce this substance, the nerve fibers begin regrowing. I don't know if you can appreciate it on this slide, but there are no nerve fibers here, and here they're beginning to regrow these green lines. And also that we also see the protection of nerve cells here in the eye. So here are nerve cells normally in the eye. After the damage, the nerve cells have become much less frequent. There's a decreased percentage of them. But with this treatment, suddenly the nerve cells are being protected, and they're not dying off. So that's just to give you a little taste of the type of things that we're looking into. And we hope to, keep the momentum going. We have a number of different things that we're focusing on, developing new blood tests and spinal fluid tests to aid in diagnosis, to predict what drugs a patient may be most likely to respond to. Maybe we could get a sense of what the future clinical course might be. We have a lot of data on this that we're actually going through right now. And once again, I think it's very important that we begin focusing, in addition to relapsing remitting MS, on progressive MS, because there's not as many options for patients with progressive MS. And I'm trying to start a large study through the VA. I'm going to a meeting next month, and we'll see how that goes. But the MS Society has recognized that this is important, and they are starting a new initiative for more research in progressive MS. And of course, everyone's interested in repair and stimulating the myelin to regrow, as well as nerve fibers.